Hello all, I'm going to do a series of video lectures here from our Chapter 6 content and then the first part of some of our Chapter 7 content to help us stay on schedule after missing an entire week of school. Plus we've got that professional development day on Tuesday the 18th when we miss yet another class. Oops, I see my PowerPoint's going to give me some problems here. All right, there we go. So in Chapter 6, and your textbook is excellent with Chapter 6. You know, what we're talking about with the class is going to line up really well with your book. So I'm going to try not to belabor any of these topics. Please keep up with your e-textbook reading and take good notes from your textbook reading as well, because the coverage in Chapter 6 is really good. Now, as we get into Chapter 7 gets a little more hairy for most students, and there are some details in Chapter 7, especially when we start talking about cellular respiration and fermentation. There are some chemistry details in there. Now, your book's not real heavy duty on chemistry, but there are some details in there that we're not going to cover, since many students in microbiology here have not yet taken a chemistry class. So I will be modifying Chapter 7 to some degree. But Chapter 6, you can use your e-textbook. It's excellent for that chapter. And same thing with Chapter 9. As we move toward the end of Unit 2, that's an excellent chapter as well. All right, so our next topic, we're talking about what it takes for microbes to grow, and we introduced the concept of metabolism, these chemical reactions that cells use for obtaining their energy and their building blocks, and then putting those building blocks together to make all of the important cell parts that you need to grow and reproduce. All right, so obviously microbes have to feed just like any other living thing. That's one of the properties of living things, is that they absorb chemical nutrients from their surroundings. And all right, so obviously if you're a little single-celled microorganism, you don't have a digestive system. You're just a single cell. Well, nevertheless, you still need to transport nutrients inside the cell. All right, and so how do microbes do this? Well, they really do this using many of the same processes that the individual cells in your body use, and which you learn about in earlier biology courses. So transporting substances, as your e-textbook discusses, it relies on the natural motion of atoms and molecules. Atoms and molecules are not rigid structures. They're actually constantly in motion. They're kind of randomly wiggling and jiggling around and bumping into each other. And that motion increases with higher temperature. It decreases with lower temperature. And that's what we actually, when you're perceiving temperature differences, heat and cold and so forth, what you're actually sensing is the greater or the lesser motion of atoms and molecules. Diffusion, a really important concept in biology. We've already talked about that before, so I'm not going to rehash it again. Diffusion refers to, and it's based on this random motion of substances that naturally occurs, but because of that, substances appear like they're deliberately moving from areas of high concentration to low concentration, and that's the easy way to remember diffusion. And we've already talked about osmosis before. That's a special case in biology. When we use the term osmosis, we're talking about the movement of water molecules across a cell membrane. Okay, so in your textbook, I would review, this figure came directly out of your textbook, and we covered a very similar figure during Unit 1, where I showed you a beaker of pure water, and then you've got a sac that's covered with a selectively permeable membrane mimicking a cell membrane, and the fluid inside the sac in red or pink, those are solute molecules of some type. It could be sodium, could be glucose, could be an amino acid, whatever it is. And we've already talked about how if this is a selectively permeable membrane that allows water molecules to pass across but not those solutes, what's going to happen? The water molecules are attracted to those water-loving solutes, and they're going to move across the membrane so that they can interact and surround those solutes, and that little sac in there is going to take up water. So as we've talked about before, with osmosis, water follows solutes. Most solutes in biology, these dissolved substances, are hydrophilic. They love water, and water is very strongly attracted to them. It interacts with them very well. So if you have a greater concentration of solutes on one side of a cell membrane versus another, then the water is going to flow across. It's going to get sucked across the cell membrane towards where you have more solutes. You guys can review this figure in your textbook as well. It's very similar to what we talked about in Unit 1. And again, I'm not going to rehash all of this, but do review what occurs when a cell is in an isotonic versus a hypotonic versus a hypertonic solution. And remember, the tonicity terms, or the tonic terms there, are referring to solute concentrations. So for example, if a cell is sitting in a hypotonic fluid, hypo means low. That means it is surrounded by a fluid in which the solute concentration is lower than it is inside a cell. So what's going to happen? Water is going to be attracted, as they're showing you here on the diagram. Inside the cell, water is going to be sucked towards where you have more solutes. If a cell is in a hypertonic solution, the opposite occurs. You have a greater concentration of those water-loving solutes outside of the cell, and so the water molecules are going to be sucked out by osmosis into the surrounding fluids. And in that case, the poor little cell is going to shrivel up, as they're showing you there in the diagram. And don't forget as well, your textbook, we looked at a similar diagram in class. Notice when you have a bacillus-shaped bacterium here, the cell wall is rigid and it really does not change shapes very much. When you have eukaryotic cells like our cells that don't have a rigid cell wall protecting them from the forces of osmosis, it's much easier for those cells to burst, like they're showing you here, if they take up water. So that can be very detrimental for them. Okay, all cells need water, so movement across cell membranes is very important, and osmosis is what dictates that. Now, how about other substances? Cell membranes are selectively permeable, as we've talked about before. So most of the important nutrients that cells need do not freely move across cell membranes. All right, now some do. All right, first of all, your textbook is dividing up the processes that microbes use to move things across their cell membranes into passive processes and active processes. And all passive means is that the substances are going to flow across the cell membrane the way they naturally want to do by diffusion. Okay? So diffusion occurs. Might need to change my pen color there so it will show up a little bit better on the black background. Active means substances are flowing against diffusion. They have to be pumped, and you got to have energy input. It takes energy to make that happen. Okay, so a couple of passive processes shown here on this diagram from your textbook. Simple diffusion, like we were just talking about. There are some substances that can freely move across a cell membrane. They can pass through the phospholipid bilayer. And so that's going to include things like hydrophobic molecules, lipids. Many of those can freely pass across a cell membrane. So like down here, we've got a cell membrane diagram. Remember, here in the center, you've got fatty acid tails. Those are hydrophobic. So hydrophobic substances like other hydrophobic substances. So they can worm their way through the phospholipid bilayer. They don't mind the fatty acid tails that are in there. Some other things, things like oxygen gas, carbon dioxide, those are small molecules that don't have any kind of a charge associated with them. And likewise, those types of substances can freely diffuse across cell membranes. Water can too, even though that sounds counterintuitive. We talked about how water molecules, there are so many of them, and they are able to squeeze their way back and forth across cell membranes, even though that sounds kind of counterintuitive based on what we've already been talking about. Okay, now what about facilitated diffusion? All right, so if you look at this diagram over here, these little squares, that's some sort of a hydrophilic, water-loving substance. And that includes ions and other hydrophilic solutes. So things like, glucose, amino acids, anything like that. If it is more concentrated outside than inside, it naturally wants to flow from outside to inside. Now those substances are water loving, so they can't just naturally diffuse across the phospholipid bilayer. Instead, how are they going to get across? They're going to mainly get across, or they're going to get across with the assistance of transporter proteins, like you see here. Proteins are very diverse. So many of the proteins that end up in cell membranes have special jobs, and those jobs are to allow certain hydrophilic, water-loving solutes to pass across the cell membrane when needed. And they're generally very specific, like this could be a protein that is transporting some amino acid from one side of the cell membrane to another. So it's going to let that pass across, but it won't let anything else. Or when you guys take Biology 201, you learn about sodium channels, potassium channels, things like that. Many of those channel type proteins in cell membranes are very specific about what they let across as well. So in this diagram again, you've got in purple here, you've got some hydrophilic solutes of some type, and what it's showing you is this protein very specifically captures those solutes and then dumps them over on the other side of the cell membrane. That's called facilitated diffusion. The substances are still flowing the way they naturally would, from where they're more concentrated to less concentrated, but they need some help to facilitate them getting across the cell membrane. All right, so active processes, on the other hand, require energy input to take place. All right, so active requires energy. These types of processes are going to be used, like you see here on the diagram. Notice you've already got a greater concentration of this particular solute that kind of looks like a thumbtack there on the diagram. It's more concentrated inside than outside the cell, but the cell still needs more of this. So this is very common for things like glucose molecules that you need to hoard for energy, or amino acids that you're going to use to build your proteins. So even though you've already got a whole bunch inside the cell knows it's going to need a whole lot more, so it's going to continue to take up these substances. So, all right, you can't just open a channel and allow these molecules to flow across by diffusion, because which way would they go? More concentrated in here, less concentrated out here. If you just opened a passageway, they would flow out instead of in. So instead, we have to force these molecules to pass across the cell membrane against their concentration gradient. And this is where active transport comes in. So this molecule here, this protein in purple, that's another membrane protein. And it's going to capture the solute and force it inside the cell. That requires energy. Remember, our ATP molecules are our special molecules that provide energy to cells. So the cell is going to have to use some ATP or some other type of energy to force that pumping to take place. That little thing that looks like a sunburst there is supposed to represent energy on the diagram. Okay, your textbook also talks about some other processes that some microbes use to transport substances across the cell membrane. For example, here's one called group translocation, where you've got a membrane protein that is capturing a solute. Energy is required to force these molecules inside the cell, and then they're getting modified. They're getting changed in some way into a usable form before they enter the inside of the cell over here. So that takes energy. That's called group translocation. Eukaryotes can do something called bulk transport. Again, this is a eukaryotic process. This is not a prokaryotic type process. And these are processes that occur at the cell membrane that engulf or take in large particles. So big molecules like proteins, or big polysaccharides and so forth, those are too big to be transported through a protein molecule that's sitting in a cell membrane. You know, a membrane protein is going to be about the same size as some big protein that's outside the cell, so it's not going to be able to allow another big molecule like that to be transported across. But eukaryotes do have a way for capturing and moving across bigger particles like proteins or even whole cells in some cases. One of those processes is called phagocytosis. This is used by white blood cells whose job it is to engulf things that are foreign to the body like bacteria and viruses and so forth and take them inside the cell and digest them. So you've probably learned a little bit about phagocytosis in your A&P classes. So what we see here is you've got a specialized cell that is attaching to some bacteria, engulfing those, bringing them across the cell membrane into a membrane-covered compartment. And that takes energy, ATP, to make that happen. You're doing some work there, engulfing those bacteria. And then those poor little bacteria are going to get digested by digestive enzymes that are inside the cell. That's phagocytosis. Pinocytosis that you see over here, some eukaryotic cells will randomly sample the extracellular fluids, the fluids outside the cell that contain various solutes inside them, and will bring in some of those extracellular fluids inside a membrane-covered compartment. That's what a vesicle is, a membrane-covered compartment. That's called pinocytosis. Some people call this cell drinking. Some people call this process over here cell eating. Okay, interestingly, this diagram that you see over here in green, that is a Trichomonas vaginalis protozoan, and it is phagocytizing a yeast cell, like a Candida yeast. So you can see, so the cells that are capable of doing phagocytosis can sometimes take in whole cells or big particles that are, you know, almost as big as they are. So that poor little yeast cell is going to get digested and devoured by the mean old Trichomonas vaginalis. So again, the processes you see here on this page are eukaryotic processes. Bacteria do not do those. Okay, and again, why do microbes and other types of cells have to eat? Well, you've got to take in nutrients, these essential nutrients we were talking about when we first started the unit, to support your metabolism. And metabolism, again, refers to, it's a combination of all the chemical reactions that take place inside a cell, catabolic and anabolic. What does catabolism refer to? Catabolism refers to breakdown reactions or decomposition reactions. Anabolism, building or synthesis. So again, catabolism, breakdown, digestion, decomposition is another term for that. You put all these chemical reactions that take place inside a cell together and you have the cell's metabolism. Okay, so we talked about different nutritional and energy groups for organisms on the planet. And if you guys remember, we and the microbes we're studying are chemoheterotrophs. That means that we use chemical nutrients both for our energy and as a source of carbon-containing nutrients or building blocks. So the general pathway is, all right, you got some food molecules up here. So these can be things like proteins and carbohydrates and lipids, all those good things. Cells are going to use catabolic reactions to break those bigger molecules into smaller building blocks. Notice as they do that, when you break bigger molecules into smaller pieces, you're breaking those chemical bonds. That gives off energy. Catabolic reactions generally release energy. Some of that energy gets lost as heat. That's what the lost heat is there for on the diagram. Okay, so once you have all these little smaller carbon-containing building blocks, those can be used for biosynthesis. Those are your anabolic type reactions. That's what they're showing you over here. So you're going to take those individual amino acids to make your own proteins. You're going to take sugars to build your own carbohydrates. You're going to take fatty acids and glycerol to make your own triglyceride type lipids. So you harness those types of things from the environment, and then you put them together in your own combinations to make all of your own cell parts. Anything that involves building or synthesis, that's anabolism. Those are anabolic reactions. It takes energy to build stuff, just like if you were building a building. It takes energy, work input to make that happen. So cells need energy to drive those processes. Where are they getting this energy from? They're getting their energy originally by breaking down their food molecules. As you break those bonds between the atoms in a molecule, there's energy stored in those bonds. Living things harness that energy and store it, usually in the form of ATP. And then when they need to use that energy for things that require energy, such as biosynthesis over here, you're going to break down those ATP fuel molecules to supply that energy. So you have this constant cycling that's taken place between these catabolic and anabolic chemical reactions that allow living things to survive. If your metabolism is working, you're going to grow. You're going to be able to reproduce. If you're a pathogen, like the microbes that we mainly focus on in this class, you're going to be more successful at causing disease. If you can't grow and reproduce inside the human body and increase your numbers, you're not going to be able to cause an infection. You're not going to cause harm to anyone. All right, I'm going to stop our first little video clip here, and then I'm going to move on and start talking about the environmental or physical factors that can play a role in the growth of microorganisms.